This is lecture one of our microbiology course. This is Introduction to Microbiology, kind of a brief introduction of some of the history of microbiology, some of the important breakthroughs of microbiology throughout the past, some of the big stepping stones of taking big advancements through microbiology, some of the little bit of future things that microbiology has in store for us, and a little bit of a kind of an overall what microbiology is introduction. So let's go ahead and get this started with the introduction of what is microbiology. So microbiology is the study of things that you can't see. The word biology meaning the study of life, the word micro meaning small, so the study of small, tiny, microscopic living things. So pretty much anything that you can't see with your eyes just looking at it. So there are tons and tons of microscopic things out there that are included in the field of microbiology. You have fungi. Over here you see yeast, and this is budding yeast over here. You have bacteria. Bacteria down here, this is E. coli, or a very similar species. You have algae, they're eukaryotic. We'll talk about what that means later. This is Volvox down here. You have viruses. There's a virus right here. You have prions. I don't have a picture of a prion. I think that's it's a type of algae up here. And then you have helminths, which are a type of worms, and they're quite a few other tiny, tiny little microscopic living organisms out there that are lumped into the field of microbiology. So, given that the field of microbiology is such a vast field of study, you have multicellular eukaryotic things, tiny, tiny non-cellular things that you can't see even with the microscopes that most labs have. Most microbiologists will pick one of these fields of studies. So you have a mycologist that studies just fungi, or a bacteriologist, a phycologist, a virologist, and so on and so forth. Just because there's just so much to talk about and so much to know. So they know a lot about, or a little bit about a lot of these, but a lot about, well, maybe one or two of them. So why do we really need to worry about microbiology? Why do we study it? Why is it interesting to us, and things like that? So throughout the history of time, microbes have killed way more people just from disease and things like that than have ever died on the field of battle for every human war throughout the history of time. So you guys can see down here, this is a Civil War regiment. They had brigades or so on, larger regiments and things in the Civil War that were about 20 or 30,000 men. And it was estimated that about 3,000 men would die on combat from bullet wounds and so forth. 5,000 men would be injured, and about 8,000 men would die never setting foot on a battlefield from disease alone. That's quite a quite a high death rate. So you had a better chance of dying from disease in camp than you did getting shot on the battlefield. So diseases were a big deal. They were easy to get, very hard to treat. We didn't understand how they worked. We didn't understand what caused them, let alone how to prevent them. So it was very easy for diseases that we can treat nowadays very easily, very easy for them back in the days to just spread very quickly, infect a large amount of people, and cause large amounts of death. So you guys can see up here, this is a picture from the 1500s. This is a tapestry from the Turks, depicting the Mongolians launching bodies of plague victims over the walls of a castle that they're trying to take over. So the bodies of the dead people from the plague would fly over the castle walls and explode in the city, spreading plague bacteria inside of the city and infecting everyone. Eventually everyone would die, and the Mongols would just be able to walk in through the front doors and take the city over. So microbes have been used for warfare just unknowingly. They didn't know what was going on. They just knew it worked. But they've been used for warfare to kill people for thousands of years, disease and all that kind of stuff. Pretty much changed the course of history. Things like the bubonic plague. The Mongolians actually brought the bubonic plague with them as they tried to conquer the world to Europe. So all kinds of crazy things have happened throughout history with microbes. But those microbes that killed thousands and thousands of people back in the day as humans have advanced in technology and society and civilizations have figured out how to deal with things like septic tanks and sewage, garbage, waste, and things like that. We've gotten a lot better at keeping bacteria that would once wreak havoc on mass cities and large amounts of people, kind of keeping them at bay from us from the get-go. So we don't really have to deal with things like cholera running in the streets from a sewage runoff and things from people just throwing their raw sewage out in the streets anymore. We have vaccines, we have antibiotics. So when somebody gets an infection from a wound down here where you say you trip and fall in camp or something and you cut your leg open, that you're not going to die from that anymore. So microbiology's done quite a lot of good things to keep us from living, whereas we're going to die from infections and all kinds of crazy things, or getting castles besieged from the plague victims, to where we no longer have to worry about the plague at all. And if you do fall and cut yourself, you're probably going to be okay if you do get an infection. So a couple of the other big things that microbiology has done for humanity. We would not even be in the United States or even pretty much be around the world if it were not for bacteria and yeast and things like that in the first place. So what does a boat have to do with microbiology, you might ask? Well, back in the day, you had to get from place to place, especially continent to continent, on a boat. They didn't have airplane jet, as you guys probably figured that out. It took months to get from Europe to the United States on a boat like this. And you had to bring water with you. You can't drink salt water. So what they would do is they would take big giant wooden barrels of water, lots and lots of those, 55-gallon barrels of water, and fill up this hull of the boat with barrels of water. Sounds like a great idea for about a week and a half until the water gets nasty and gross from the fungus and stuff growing inside of the barrels. The water will become stagnant and will become toxic and it's unsafe to drink. So how do you get around this problem? It takes months to get there. You can only keep a couple of weeks worth of water. You take lots and lots and lots of beer. That's how you get around this. So what they would do is they would take beer with them. Pretty much twice the amount of barrels of beer as they had of water. And this wasn't just your average old beer that you could go get at the gas station. This was beer that you had to have a liquor license to sell, and could only get at the liquor store that was like 40 or 50% liquor, 40 or 50% alcohol. Very strong beer. And what they would do is they would take 50% beer, 50% water, or 25% water, depending on the day, and mix them together. The alcohol would kill off the bad stuff in the water. The water would dilute the beer down to where it wouldn't get you sloshed. And that's how they got from place to place. So the beer would make the water safe to drink. The beer you could carry in barrels because the alcohol killed everything. And then when you diluted them together, you could make stuff last twice as long. So that's pretty much how people got from place to place if they had to take water with them from a long place for periods of time. In fact, this was so common back in the day, water was so dangerous to drink that they would usually give children back in the Roman days, and even up until the 1500s, they would give children very, very, very low alcoholic strength beer, maybe 2 to 3%, maybe a beer that you might get at the gas station nowadays to children, because it was kind of risky to give them pure water. It was possibly going to be contaminated. It could have had things in it that you couldn't see. They didn't know what it was. They just knew that it was dangerous. They knew that if they gave them beer that you were probably not going to get sick. So even little kids had low alcoholic beer. Another thing here, the air that you breathe. Some of you guys may know this band. Some of you guys may know Crosby, Stills, Nash and Young, the band that Graham Nash came out of and joined later on. This was his first band. Well, anyway, the air that you breathe, about 40 to 60% of the air that you breathe actually comes from bacteria, not plants. So bacteria make lots and lots and lots of oxygen for us that we breathe. They live in the oceans, they live in ponds and things like that. And they carry out a very primitive form of photosynthesis. So I just mentioned alcohol and beer, bread, lots and lots and lots of other food things like cheese and things come from bacteria. Essentially, cheese and yogurt is just bacteria poop. And then not to mention the countless deaths throughout history that people have just died of a horrible disease in the face of history that's been changed from things like the Black Plague. You guys know the nursery rhyme, Ring Around the Rosies, Paga Full of Posies, was written about the Black Plague. So even things like that carry on into modern day times. And then the last one up here is vaccines. So lots and lots and lots of vaccines, antibiotics and things are produced by fungus, other bacteria that humans have learned to harness and figured out how to tweak and manipulate for use for humans. They just figure it's like we figured out how to use the products that bacteria and fungus produce to help humanity, which is pretty neat. So we're going to start talking about microbiology, kind of what it is. And kind of the first thing to introduce microbiology after what it is some of the big fields of microbiology that are found today. There's quite a few of them, and each of them kind of specialize in different focuses of the microbiology world. Since it is so broad, there's so many things to study. There's tons of different things to do with it. There's lots of different fields to focus on. So the very first one is public health and epidemiology. So this is kind of a big deal at the current moment, given all the facts of we have mass widespread travel, all the concern about diseases at the borders, coming across borders and things like that, international borders. So if you decide to go into a field that deals with public health or epidemiology, your job will be to look at diseases that maybe are localized outbreaks in your community. Maybe your community has an STD issue. Your job is to figure out where that comes from, or maybe your community has a high disease rate of a particular viral disease or something like that. Maybe a new disease that's shown up in your area, and your job is to figure out where that comes from, how to control it, and things like that. The next one would be biotechnology. And this is using living things, particularly in our case would be yeast, bacteria, and things to do something for humans. So you're going to put them to work, per se. So you take yeast, you put them to work making alcohol, brewing beer, or brewing things like alcohol for vodka use, distilling, and things like that. So you put organisms to work for humans. And your job would be to manage the yeast cultures at the brewery, manage the stem cells and the bacteria that are used to manipulate genes and things in labs. You have environmental microbiology, excuse me. An environmental microbiologist is someone who deals with microbes in the environment, as the name implies. So your job would be to go out in places that are healthy and to figure out what a normal, healthy environment looks like. What no humans have contaminated it with, and things like that. And then your job would be to go in and look at messed up environments that humans have impacted. So your job would be to look at the bacteria that are in the healthy environment and things, and then go look at the bacteria that are in the unhealthy, human-impacted environment, and figure out what is changed. How can we get it back to what it needs to be to make this environment healthy again? Why are the trees not healthy? Why are the animals not healthy? What's happened because of these humans? And the other way you can look at is what have the bacteria done to the environment? Sometimes we cause sewage runoff, which causes a massive overgrowth of E. coli, which can kill large amounts of fish and things like that. So your job would be to figure out how to fix things like that. Immunology. And this is someone who uses the immune system or bacteria and things like that to figure out how to make vaccines, how to make new tests for allergies and things like that. You have genetic engineering. This would be someone who takes the DNA of bacteria, the DNA of fungus, and alters it in a lab to figure out how to make them make new things, or make new species of organisms in a lab that do new and novel things for humanity. So you could take a bacteria and insert a new gene to make them make things like insulin, per se. And that's how we make insulin for diabetics. Bacteria produce it. They used to get it from pig pancreases. You have agricultural microbiology. And this would be dealing with diseases that plants and things like chickens and cows and pigs and things that humans eat would get, or are working at like a dairy, keeping foods the cattle healthy, and making sure that their udders and things don't get disease and things like that. One of the other big things of research that's coming up now is a resistance for microbiology in these food organisms. For diseases and things, the overuse of antibiotics in these food or animals has caused the microbes that are common in those organisms to become resistant to very common antibiotics. So it's kind of a big deal at the current moment. And then the last one is food microbiology. This is probably the most common field of microbiology outside of research and things like that. So every single food plant on the planet, if they make something that you eat or something that maybe your pets eat, they're going to have a microbiologist that works on staff, probably more than one of them. And their job is to go around after the plants close and take throughout the day. They'll go around and swab blades and things that cut meat, or grinders and things, ovens, and take random samples of the food or the product that's being made or whatever it is, if it's dog food or human food or whatever it is, cat food, and make sure that there's no microbes that are found in there that there aren't supposed to be there. You don't want bad bacteria in these things after the processing and cooking and all that stuff. You want things to be clean and sterile, and you want the surfaces that are touching these food things to be clean. So your job would be to walk around and swab surfaces, grow cultures, and make sure that the food that you and I, as well as your family and friends, as well as probably your family and friends and your pets, are eating stays safe and free of contamination. So let's take a step back in history to kind of the discovery of bacteria, the kind of discovery of microbes overall, kind of how they were discovered, kind of how people came to understand that bacteria existed. So it's hard to take a step back for you and I to imagine a time in history where bacteria and the knowledge that they existed just wasn't there. To imagine that something you don't know something that you and I are aware of. But you have to put yourself in the shoes of someone who lives in the 14 or 1500s that is unaware that something that they can't see exists. And it makes sense if you think about it. I can't see it. They don't have microscopes. They don't have magnifying glasses. They don't really even have glass, for that matter, to make windows and things out of that's really clear they can see through yet. So they're not really able to make things that can help them see stuff they can't see. So what I can see is what exists. There's nothing out there that's smaller than what I can see that exists. That's just kind of their mindset. We have to accept that. So anyway, once the technology started to become available to make microscopes, that glass started to become clear and useful and things like that, people started to notice that it had a magnifying ability. So they started to make microscopes and things like that with it. So once that started to occur, people started to look at things under these magnifying microscopes and things. So people started to draw what they saw and things like that. Sent around all these different papers and stuff and talking about it. And this is kind of the birth of microbiology. Well, anyway, one of the first people to do this was a guy named Robert Hooke. He's an English guy that really just liked to look at stuff underneath a microscope. He would just pretty much grab anything he could find, living or dead, fake or human-made or natural, and throw it underneath his microscope and look at it. And he put together a huge book called Micrography. It's all hand-drawn pictures of things underneath the microscope, and they're really, really, really detailed. He drew a fly head that looks almost identical to what you guys would see today underneath a really, really modern microscope. This was in the 1500s. So one of the things that he's credited for, and the most famous things that he did, was he took a piece of cork. Cork comes from the bark of a cork oak tree. It's just cork like a wine cork or maybe a cork coaster. So he just took a small sliver of that, and he put it underneath his microscope, and this is what he saw. So you guys can see these little squares here, and these are the cells of a plant, and this is the plant cell wall. The plant cell would be inside, the plant's died, and the cell has died, and it's evaporated and left behind just its little hard cell wall, and that's what you're looking at. So he was the first person to ever see anything like this underneath a microscope, or first person to ever see anything like this at all, for that matter. So he looks at these things and he goes, these things look like little tiny rooms that look very similar to where the monks live in the monastery near my house. Everything you need in one little room. The guy lives in there. He's got everything he needs for him to live. And these kind of look very similar to that. Everything you might need in one little square, sectioned off, little equal, even little squares and rooms and things. These are called cells where the monks live. Prison cells are the same thing. These are kind of similar. Why don't I call these things cells? And that's where the term cell comes from. Little tiny room has everything you need. Robert Hooke put these two together and coined these as cells. And that's where the term comes from. He didn't know what he was looking at. He didn't know that they were cells, what they were. He just called these little things cells. He didn't know that they were the root of all living things that we do now. He just knew that they were some sort of little sectioned off thing that he could observe. Well, anyway, move on a little forward to the very first person to actually observe a living organism underneath a microscope. So this guy right here, Antony von Leeuwenhoek. So the problem with Robert Hooke's microscopes is they weren't very good. They were good, but they were okay. They weren't even really good. They were okay. Antony von Leeuwenhoek, on the other hand, was really, really, really wealthy. He was not a scientist. He was just a merchant. He had a lot of money and a lot of free time. And he liked to tinker and make things. And he was really determined to make really good microscopes. So he had a lot of money that he could use to buy really good, really pure glass that would allow him to make very, very, very high quality microscopes. So you see the little glass lens here. You put the specimen right here on the tip of this little needle. You hold this little thing with your thumb and you hold it up to the light. And you hold, this is the back of it over here. This is the front. You would look through this side and hold this up to the light, and the little specimen would be illuminated, and you could look through and see it magnified. So what he did is he made a really, really, really good microscope, and his microscope was able to see about 5 or 10 times better than the microscopes at the time. So he was out in the woods one day, out fishing one day, taking a boat ride, and he took a drop of water out of the lake, and he put it on the tip of his little microscope, and he held it up to the light, and he looked at it. And what he saw were these little things. He called them animalcules. He wrote about them, he drew them, he wrote how they moved, he was able to see their colors, everything about them inside of them, very detailed. Microscopes at the time were not good enough to be able to see this. He was the only person that had a microscope that was good enough to see this. So keep that in mind when we move forward. So he would write about how these things moved. He saw them eat. He saw them swim, he saw them grow, he saw them divide, and he wrote extensive notes about them. So you can see animalcules down here. This is his actual drawings. In fact, he's so famous that Google gave him a doodle on his birthday a couple of years ago. So anyway, what happened is he wrote this article. He sent it to the Royal Society of London, saying, hey, look what I found. You guys might want to go look. So they went out to look and say, oh, this is cool. We might want to go see if they're there. And they couldn't find them. Their microscopes were terrible. They couldn't see anything moving. They couldn't see anything. Oh, this guy's crazy. There's nothing there. So he keeps writing. He keeps writing. He keeps writing. They keep checking. Nothing's there. Eventually, they quit. They start ignoring his letters. And then all of a sudden, microscopes in England get good enough to see tiny little things. They check again after he keeps persisting. They finally see them, and then they say, oh, he was right all along, and they invite him over, and he joins the Royal Society of London. So that's kind of how that always works. So you were wrong, you're wrong, you're wrong, oh, you were right. Come on, welcome, join aboard. So anyway, another big step to move forward in the world of science was figuring out where life itself comes from. And this is not the question of where life came from in the very first sense of life. This is figuring out where life comes from at the current moment, where you and I, where do our babies come from? How do they get here and things like that? So at the time, it was a very common belief that life could come from non-living organisms, or non-living material. Imagine you would have a piece of meat out on your front porch, or a dead animal or something like that, maybe you found on the side of the road back in the day, and you don't know where life comes from. You're not around to see flies land on this dead organism, but the next time you walk by it, it's just covered in maggots. And your brain, not understanding what's happening, goes, those maggots just poofed out of existence of that dead organism. Life just came to existence from some force within inside that dead carcass, that dead meat just brought forth these maggots. And another example that's often used is mice and things, rats coming forth from stored grains, bags of wheat and things. You're just sitting home one day and all of a sudden a rat just burst out of the corn bag you've got sitting in the corner. And you didn't see the rat go in there, so you don't know that he crawled in there obviously earlier. But all you know is that he just poofed out of there. So that was a pretty common belief that life could come from non-living organisms or non-living material. So this guy right here, Francisco Redi, decided that he was going to show everybody that they were crazy, and put this experiment, up with this experiment to put the belief in spontaneous generation, life from non-life to death. So anyway, what he did was very simple. He got 3 jars, 1, 2, 3, 3 glass jars, and he put a steak or some meat inside of each jar. In one jar he left open to the environment. One jar he covered with gauze. Now, if you guys know anything about gauze, it's not totally airproof. The air can go through it, but not a lot of other things are going to be able to go through it, especially flies, which is what he was looking for. He was trying to see where maggots come from. And the last jar he covered with parchment. Parchment has no holes in it at all, and it is airtight. Air cannot go through it. That's the key here. So he let these things sit out for a couple of days, and he watched them pretty much 24/7. The open jar, the flies would come in, the flies would land on the steak. He watched the flies lay eggs. Eventually, he watched the eggs hatch into maggots, watched the flies fly away. The maggots would develop, the flies into flies, and then the adult flies would fly away, watching the steak go away. And the whole process he watched happen. The gauze jar, the flies would be attracted to the scent of the decaying meat. They can't get to the meat. They will land on the surface of the gauze and lay their eggs. He watched this happen. He watched the flies, the maggots never develop from the meat itself, but he watched them develop on the top of the jar. They never got to the meat. They never came from the meat. They came from the flies. This was the link that he needed right there. This one was really helpful, but this one was the one that he needed to convince everybody that they come from the flies. Because you could still say they come from the meat here. The flies are just a coincidence. This is what he needed. And the last one that he needed, or that he did, was to show that even with, if you get rid of the flies completely, no flies show up at all, there will be no maggots. So he covered it with parchment paper that's sealed to the air. No air can get in or out. No smell gets out, so the flies are not attracted. No flies, no eggs, no maggots. The steak is never eaten by maggots. This convinced people, oh, you have to have the flies to have the maggots show up. Life does not come from non-living things. If it did, this steak would have developed maggots every single time, but it doesn't. But there's a problem with his experiment. It's airtight, yes, but in the air was tons of things that they didn't know existed, like bacteria, mold spores, and lots of other tiny little things that they had no earthly idea were out there. So even though this thing is closed to the air and the maggots didn't get in, bacteria and all kinds of stuff that were inside of this jar still contaminated this meat. So even though the belief that large things cannot come from non-living matter, that flies and maggots and maybe rats and things don't come from non-living things, that bacteria, these tiny little microscopic organisms that we can obviously see exist, that they're kind of starting to understand are out there, do. They just poof into existence from that meat. So even though the idea of spontaneous generation went away, the idea of spontaneous microbial generation quickly replaced it. That the idea that bacteria and fungus and things, they didn't really know what they were, they didn't understand them yet, but that those type organisms would just poof out of existence. They're too small to see, you don't know where they come from, they just show up on rotting meat, even though you block the air out. I don't know where they came from, they obviously came from the meat. So very quickly after that showed up, people started to try to disprove that bacteria themselves came from nothing. People didn't really believe that. A lot of scientists were of the belief that life had to come from something else that was living. So they tried to do their best to figure out how to show that bacteria themselves, these things that just were out there, that they didn't understand yet, had to come from something else as well. So one of the first things that they figured out very quickly, and people had known this for a long time, is if you boiled water and boiled foods, it would make it safe to drink. If you let it sit out, it would get funky and dirty, and it would make it unsafe to drink. So they knew this for a long time. So one of the very first ways to try to prove that spontaneous microbial generation wasn't a thing was they were going to boil some broth, some beef broth and stuff, and let the bacteria die inside of it, make sure they were all dead, and then they would show back up if they were in the beef broth. They didn't know they were in the air. So this guy boiled his, John Needham, he boiled his experiment, boiled his flask, let it sit. They had bacteria fell in from the air and recontaminated his experiments. This didn't disprove it. As time moved forward, people figured out that you kind of needed to seal the jars. And this guy right here, that figured out very quickly that if you left them open, they would get stuff inside of them. But if you left them closed, that they wouldn't. And that kind of led people to the idea that stuff may be in the air. You could show that if you left it open and you didn't boil it at the first place, or you did boil it and you left it open, it would still turn the same. But if you boiled it and plugged it and it wasn't able to get air inside, nothing would happen to it. So people were kind of getting the idea at this point. And then this guy comes along, Louis Pasteur. And he decides that he's going to come up with a way to put this idea to bed over once and for all, this idea of spontaneous microbial generation. And we did quite a lot of things that are very important. This one being the first one that we're going to talk about. And a couple of other things. He made a couple of very important vaccines. But this one we're going to talk about is the most important one for our class, other than pasteurization, which we'll talk about later. So, um, anyway, it took 200 years for them to move from Francisco Redi showing that life doesn't come from non-life for Louis Pasteur to show up to get rid of this. 1861, right around the outbreak of the American Civil War, Louis Pasteur publishes his very first paper showing that bacteria come from the air. And he put together a very unique experiment to show that they came from the air. And that was the problem that the earlier people hadn't done. They hadn't shown that bacteria come from the air yet by proof. They hadn't proved that bacteria were in the air. They just thought that something was in the air. They didn't know what it was yet. So Louis Pasteur proved that it was bacteria, that stuff was in the air. So one of the first things that he did was very interesting. He took essentially a toilet paper tube and he stuffed it with cotton, a little filter, and he sucked it through with his mouth to get a bunch of air through. And he took that little cotton plug and he looked at it underneath a microscope. And he drew what he saw, all the mold spores, all the little bacteria, and all the tiny things that he could see. And he drew pictures of. And then he dunked that little cotton plug in a vat of sterile broth and let the bacteria that were inside of it grow and get nasty and yucky. And then he took a sample of that and drew it again to show that the bacteria that he sucked out of the air, when you grow them, they will grow and they stay the same, and that you can make more of them, that they obviously are what's going to make this broth nasty. So that was his first step. And then what he had to do is he had to show that even if you're open to the air, that was the key here. You had to be open to the air, but you could not have the bacteria in. So even if you're open to the air, it's still the bacteria, not the air itself, that's causing the stuff to go bad. That's causing bacteria and things and mold spores and stuff to cause these steaks and things to rot. So what he did was he developed these very specialized little flask here called swan neck bottles or swan neck flask. And how this works is it's a little glass tube here that's hollow with essentially a little hollow glass straw. Now, if you guys have ever ridden a roller coaster, you know how this works. The top of the hill, the highest point, is the highest point you'll ever go. Gravity cannot push you higher than the highest point. The same concept works for air. The force of gravity and the force of air will not go higher than the highest point. So since this point over here is higher than the entrance, air pressure cannot push air any higher than here. Or excuse me, air can move, but tiny little particles that are heavier than air cannot be pushed up higher. It dissolved any bacteria floating in the air, mold spores and things like that. The air pressure itself can't push them higher than the highest point. So what happened is he boiled it and sterilized it and killed all the bacteria inside of it. And he let it sit. He let it sit and he let it sit and he let it sit for weeks and weeks and weeks and weeks. Now what happened is this thing was open to the air. Air would pass through all day long. Air would pass through. But all the bacteria and dust and mold spores that are too heavy to be pushed up this swan neck by the air pressure would get trapped in the little neck, the little U curve there. So air could go through, but the bacteria wouldn't. They couldn't go through. So that this stuff would remain sterile for hours and hours and days and weeks and months, no matter how long it set. As long as it never came in contact with the bacteria from the air, it could still come in contact with the air, just not the bacteria. It would remain sterile. And then what he did was he knocked right here, he knocked off this section of the glass, took a sample of this stuff, looked at it under his microscope, and saw it was the exact same stuff that he had earlier collected from the air and that he had grown in his sample. And then he took another one of his little flasks that he had, tipped it sideways, contaminated it with some of this dirty, nasty stuff, and let it sit back forward, and then a couple of hours later, it was nasty and contaminated and gross again. He took a sample of this stuff, compared it to what he saw here, compared it to his earlier samples, and said, oh, bacteria is in the air. I can show it because it was here. I can show it from earlier because I sucked it out of the air, and I can show it earlier from my experiments showing that this stuff links all together when you grow. This actually worked. This finally showed that even though the air's there, it has other stuff inside of the air. These living bacteria, these living organisms don't just poof into the broth. It's not just there. It comes in from the air. but it is not the air that's doing it. It's stuff that's in the air. It's the bacteria that's causing them to grow. And this showed everybody that it worked. Now, the problem with his experiment here is there's two types of broth. Beef broth that comes from essentially boiling beef, chicken, things like that, animal broth. And then there's vegetable broth or wheat-based broth, like soy broth, that comes from boiling plants. Now, beef broth is very easy to get sterilized, very easy to kill everything inside of it. Plant broth is not. It's almost impossible to get sterilized, especially back in the day. We can sterilize it now very easily, but back in the 1860s, they couldn't. It was very difficult. And they were unaware of something called an endospore that's found only in vegetable broth. It can survive the process of boiling. So essentially, it kills all the bacteria. The endospore survives it. The endospore is essentially a bulletproof bacteria. It survives the process of boiling and then starts to divide and divide and divide and grows a bunch of bacteria back up. So people thought for very quickly that, I can't replicate Louis Pasteur's results. I can boil, boil, boil. It stays sterile. All the bacteria stays out, but it still goes contaminated. It's the air. And then finally somebody figured out, this guy right here, Ferdinand Cohen, that endospores existed. He showed how they worked. He showed how to kill them. And then once the process of showing how to properly kill endospores, how to properly sterilize both types of broth arose, and that you could sit both types of broth open to the air for a really long time. As long as you kept the bacteria out, they would never go bad. It took almost 200 and some odd plus years for them to actually abandon this idea. But once they finally had, everybody finally got on board. And then quite a few advances, quite a lot of important advances in the world of microbiology took place. One little last note, one of Louis Pasteur's swan neck bottles is still sitting in the Louvre Museum in Paris, and it's still sterile from back in his original days. They haven't tilted it yet. You can go, I think it's still maybe on display, but I'm not sure about it, but I know they have one. So anyway, right after the abandonment of this theory of spontaneous microbial generation, the next about 40, 50 years or so is termed the golden age of microbiology. So people had finally figured out that bacteria were a thing. They figured out where they came from. They kind of figured out how to see them. Microscopes were getting pretty good at the point. So lots and lots and lots of scientists were working on bacteria, figuring out where they came from, how they cause disease, where disease came from, how to prevent disease, and things like that. So lots and lots and lots of advancements in that little small time period. So let's talk about some of them. So in the beginning, some of the early vaccination campaigns, things for like smallpox, polio, mumps, and things were rolled out in the 50s and 60s, 70s. Some of the mass vaccination campaigns were accompanied by the development of antibiotics for diseases that once killed tons and tons of people throughout history. But pretty much nowadays, we've gotten a lot of the diseases from history that were mass killers kind of eradicated through the uses of these vaccines, antibiotics, control of septic tanks and sewers, runoff in agricultural streams, limiting the use of antibiotics and overuse of things like that. But even though that a lot of these diseases have been cured and things like that, there's still a lot of stuff to go on for the field of microbiology. A lot of this is going to have to do with medical research in the future, developing bacteria that can produce materials that are useful for humans, for the medical fields like insulin, different types of antibiotics, as well as for food products, developing different types of bacteria that can produce different food type things. Different flavorings, maybe useful for food overall, different types of bacteria, lab-engineered meats and things. One of them is also going to be for environmental microbiology. That's a big deal already, but it's going to become even more of a deal in the future. So you guys can see down here the big BP oil spill from about 2010 or so. They dumped thousands and thousands and thousands of gallons of bacteria that had been genetically engineered to eat petroleum. So the bacteria would eat the petroleum, they would change it into a non-harmful form. The bacteria would then die, and that petroleum would then be neutralized in the ocean, which is pretty neat. So developing bacteria like that, things to mitigate human disasters, is a pretty good thing to look forward to for microbiology. So let's go ahead and talk about some of the things that, some of the first breakthroughs of microbiology, some of the first things that you guys are going to get introduced to with microbiology, kind of what it is, kind of how it operates, and kind of how to talk about it. So one of the first things to jump into with microbiology is the concept of what is a cell. So cells, I've kind of introduced you guys a little bit to Robert Hooke a little bit, the concept of a cell being a little self-contained unit of life that has everything it needs to be a living organism. So bunches and bunches and bunches of experiments had been done. You guys can see down here that what had been occurred, what people had figured out. And then these guys, Schleiden and Schwann, start to look at experiments that had been done earlier in time, that these guys way before them had figured out. They start to do a bunch of their own experiments to kind of define what a cell is, this vague idea of a cell. They're going to refine Robert Hooke's idea and kind of put words to what a cell is. So this is what is known as the cell theory. So they came up with these first three things. The rest of them down here have been added later. But these three things here were the first three things, definitions that were used to define what a cell is, to take this vague idea of tiny little microscopic things that are out there that are kind of little self-contained units of life, and kind of what it is. What is a cell? So this is the early definitions of what a cell is. So cell definition number one. If you are a cell or a living thing, you are made up of at least one cell. You may be one single cell. There are quite a few of those out there. They probably outnumber us by multicellular things by a couple of hundreds of thousands to one. You either have one cell or more than one cell. That is a living organism. If you have non-cells or if you have less than one cell, you are not a living organism. Every single cell on the planet comes from other living cells. They looked at Francisco Redi. They looked at Louis Pasteur's experiments and things like that. They looked at Francisco Redi's experiment and John Needham's experiment and things like that to see that life could not just poof into existence. And they kind of had figured this idea out already. And a lot of scientists had kind of already abandoned it. It just took a really long time for people to prove that that was a thing. They didn't have the technology to prove it yet. So that life came from other living organisms. If you are a living thing, if you are a cell, you came from some other already pre-living cell. Now that begs the question of where did the first cell come from? Either nature created the first cell or some supernatural thing created the first cell, and it doesn't really matter to me. Either one is fine. We are here either way. And the last one is the cell is the smallest fundamental living unit of life. There is nothing that is more fundamental or uniquely living than a cell. A cell has everything that you need to be considered living. If you have less than what you need to be considered living, you're obviously not alive. So cells have everything that they need to be alive. They have everything they need to make energy. They have everything they need to be able to reproduce by themselves. They have everything they need. If you're not able to reproduce by yourself, if you can't make your own energy and things like that, if you can't make your own proteins, you're not considered a living organism. Cells can do all of that. So these first three things were the very first definitions that were written down to define what a cell is. These other ones were added later. So this one is the total activity of a cell of an organism depends on all the independent activities of the cells. Easy way to say I'm 100% healthy, 100% of my cells work. I can do 100% of my normal daily activities. Hooray, my body works 100% well. If 75% of my cells are sick, on the other hand, 25% of my cells work happily, I can only do 25% of my normal activities. Makes sense. Cells are the only things that are capable of making cellular energy in the form of ATP. They are the only things that can make it. They are the only things that can use it. Nothing else on the planet can use or make ATP. Only cells. Every single cell on the planet is either going to contain DNA and, this should not say or, this should say and, DNA and RNA. Not either, both. DNA and RNA. Every single cell contains DNA and RNA. Scratch the or, the either. DNA and RNA. They didn't know about DNA or RNA in the 1830s. These were discovered in the 1940s, 50s, and 60s, so they didn't know they existed yet. And then pretty much every single cell of similarly related species is pretty much the same. And this makes sense if you think about it. If you take a cell from a dog, a cell from a fox, a cell from a coyote, a wolf, they're pretty much going to look the same because they're all canines. But if you take a cell from a dog and then a cell from a jellyfish, they're going to look a little different. So that one's pretty easy. So if you are a cell, you adhere to these characteristics. Now, these characteristics are what we use to define cells. You have to fit all of these definitions to be considered a cell. You can have all but one of them and not be considered a cell. That's how that works. It's an all or nothing thing. You can have literally all of them but be missing one of them, and you're still not considered a cell. So one of the other big advancements in the ideas of microbiology was the linking of bacteria to specific diseases, saying this particular species of bacteria causes this particular disease. And when you're able to do that, you're able to narrow down where that bacteria itself comes from, how to treat that bacteria, how to prevent that bacteria, how to specifically make targeted treatment for that particular disease. Oh, that disease is so-and-so. Well, what causes it? I don't know. Let's go figure it out. And this guy right here, Robert Koch, he's a German scientist, was the first person to figure out how to do this. The first person to figure out how to link a particular organism, a particular bacteria microbe, to a particular disease that it caused and say, this organism causes this disease. Now, his experiments are known as postulates, his ideas, collections of thoughts, how to do something, or postulates. And we still use them today to figure out how to link new diseases if we don't know what a disease is caused by, how to figure out what that disease is caused by. They still use his experiments, his ideas today to do this. So his experiments are pretty neat. So what you have to do is the first thing you have to do is if you have sick organisms and you don't know what's causing that disease, is you want to figure out what bacteria they have present in them. So what you're going to want to do is you're going to want to go take look at your diseased population and your healthy population. So if every single organism in your population is healthy or sick, all the sick organisms, if it's the same disease, should all have the same symptoms, and that makes sense. All these mice should be coughing, sneezing, have the same runny noses, whereas half of them shouldn't have hopping around, bleeding out of their ears and things like that. They should all have the exact same symptoms. And the other thing is when you look for bacteria underneath a microscope, you should only find bacteria in these sick animals. Healthy animals shouldn't have it, and that's a big deal here. If they're healthy and they have bacteria in them, this guy has the same bacteria and this guy has it too, it's obviously not that species of bacteria that's causing the problem. So you guys can see here, this guy lacks the little black rod-shaped bacteria that this guy has. So this is the difference here. Now your job as a microbiologist, Robert Koch, was to find the difference. There's millions of different species of bacteria that live on organisms. His job was to go figure out which ones are different. That takes a really long time. So all the sick organisms should have the same symptoms. They should all have the same different bacteria inside of them as well if it's the same disease. None of the healthy organisms should have symptoms. None of them should have bacteria that are found inside of the sick organisms in them either. So you take a sample from the sick organisms and you grow it in a lab. It should match this organism. It should look just like it when you put it underneath a microscope. You grow it by itself, it should look just like it. You take a sample from the healthy mouse, you grow it in a microscope in a lab, and you have no bacteria, and that proves that there's none there. Some in the bad stuff, none in the good stuff. So this thing looks exactly like this when you put them underneath the microscope, and this is how you prove that they do this. You take a sample of the bacteria from your culture plate that you got out of the bad animal. You put it in a brand new healthy organism that's never been sick before. That organism will develop, or not develop if you have the wrong species of bacteria, but if you do, it will develop symptoms. And those symptoms should be coughing, sneezing, runny eyes, the same symptoms that match the original species or the original symptoms of the mouse that you're trying to test from. This organism, you should do an autopsy or necropsy on this organism. And you will also find the same bacteria inside of it. You'll see the same bacteria that matches here, that matches here. You grow this bacteria out of this organism, back up in a lab, pure culture it out. It matches here, matches here, matches here, matches here. All of these organisms match. It's not found in any of the healthy organisms. Only this bad bacteria is found in the diseased ones, not the healthy ones. It's only found in the ones that are sick, not the good guys. All these steps show that it's the same organisms that causes the same symptoms. This is how you link. This organism causes this disease. And this is how he did this. This is called Koch's postulates. Very cool experiment. Very, very simple experiment. And we still use this today. To show a brand new disease. We don't know what species it is, what organism it is that causes it. We still use the same steps to show, oh, this is the organism that causes this. Now, this is the word form of what I just went over if you want to read it. So let's go ahead and jump into what the different types of cells are. They're very simple. So really a brief introduction. You guys probably know a little bit about this from biology in high school or maybe your last biology course. But there are two different types of cells. You have one cell called a prokaryotic cell and one cell called a eukaryotic cell. Now, what does this mean? So prokaryotic, the prefix pro means pre or before. Karyotic means nucleus. So this means an organism, a cell type that is before a nucleus. So this is one type of cell. And the other type of cell is a eukaryotic cell. Eu meaning true, karyotic meaning nucleus. So a true nucleus, a cell that has a true nucleus. So if you look up here, this is a prokaryotic cell. This is a eukaryotic cell. A nucleus is a little tiny small ball of protective lipids and membranes and things that keep the DNA of the organism safe. Prokaryotic cells don't have one. Eukaryotic cells do. That's one of the big key differences between the two types of cells. There are lots of other differences, but that's one of the big ones. Every single bacteria on the planet is considered prokaryotic. All animals are considered eukaryotic. We are eukaryotic. All bacteria that we will talk about in this class, and for the most part, all organisms that we'll talk about in this class are going to be prokaryotic. I will tell you guys when they are not. So the easiest way to tell these two things apart is obviously the presence or absence of a nucleus. But if you don't have if you don't have testing abilities, you just can't do that. Can't test for DNA, and all you have is a microscope. The easiest thing is size. You put these guys underneath the microscope. The prokaryotic cell, you guys can see, is significantly smaller than the eukaryotic cell. These guys are so much larger. That's the easiest way to tell them apart. So bacteria have been around for about 3.5 billion years on the Earth. You guys can see some of the history down here of the time history of life on Earth. So bacteria show up around here. And it takes quite a while for eukaryotic organisms to arise, about 1.5 to 2 billion years. So once again, our prokaryotes and eukaryotes. Eukaryotes have that true nucleus, a little control center around them, keeps their DNA safe and things like that. Prokaryotes don't. Eukaryotes, they're much larger. They need to protect their DNA, keep it all localized. Prokaryotes, they're so much smaller. They don't really have a lot of complex things inside of them to worry about. Their DNA is much more simplistic. They can keep it smaller. They don't really have to worry about keeping it as safe. So, a couple of characteristics of prokaryotes that are found that are unique to prokaryotes, and these are some of the characteristics that we use to define what a prokaryote is. Every single prokaryote on the planet is unicellular. Unicellular, one cell. They are all also microscopic. That is the key characteristics of prokaryotes. All of them are unicellular. Every single one of them is microscopic as well. They don't have a nucleus. They may have a little ball of DNA inside of them. DNA sticks to itself. It's called a nucleoid or a bacterial chromosome, quote-unquote chromosome. They do not have a nucleus. They don't have organelles that are bound by membranes inside of them. They don't have mitochondria. They don't have a Golgi apparatus. They don't have an endoplasmic reticulum, things like that. They don't have complex cell structures inside of them that are bound by membranes like a eukaryotic cell does. One of the other key characteristics here of a prokaryote that's only found in prokaryotes is the presence of a cell wall made out of a unique substance called peptidoglycan. So peptidoglycan, peptid meaning protein, glycan meaning sugar, so it's a unique complex of proteins and sugars. Peptidoglycan is only found in our prokaryotic cells. Only found in prokaryotes. No eukaryotic cell on the planet naturally produces peptidoglycan. Since it's only found in prokaryotes, it makes a really good target for antibiotics. and that's how penicillin works, and we'll talk about that concept later. Prokaryotic cells divide by a process called binary fission. It's asexual, very simplistic process. This is a very good example of how this works down here, the Venn diagram between the two different cell types. Essentially, you have one circle, and the circle just splits in half and creates two identical circles from earlier. So imagine one circle that just splits in half just like that, and you end up with two. Creates identical copies of the original parent cell. These guys also have a smaller ribosome inside of them called a 70S. S stands for Spedbard, which is how fast a centrifuge spins. You don't need to know that at all. It just means that their ribosome is slightly smaller than the ribosomes that are found in a eukaryote. So this is some characteristics of a eukaryote. Eukaryotes can be either unicellular or multicellular. Unicellular eukaryotes are things like yeast. Multicellular organisms that are eukaryotic are things like us, frogs, larger organisms, things like that. We have true membrane-bound organelles like our mitochondria here, endoplasmic reticulum, Golgi apparatus, and things like that, whereas our prokaryotes don't. Our cells very rarely can divide by asexual reproduction, making an identical copy of themselves in that sense. Our cells divide by a process called mitosis for our body cells, or meiosis for sex cells, things like sperm and eggs. So it's a totally different process that involves the changing of chromosomes, duplication of chromosomes, moving over, crossing over, and all that good stuff. So we have a true membrane-bound nucleus, that big deal here. Keeps all of our DNA safe, keeps it all protected and localized in one little spot. Bacteria have chromosomes that are round. They only have one chromosome. We have multiple chromosomes, and ours are not round. Ours are straight lines. Now, if eukaryotes do have cell walls, not every single eukaryotic cell has one. Animal cells don't. Fungus cells do. Their cell walls are made out of a type of sugar called chitin. It's also the same thing that makes up the exoskeleton of insects, by the way. So if you like mushrooms on your pizza, technically you're eating insect exoskeleton sugar. Now, it's not the same thing, it just chemically is. But anyway, and these cell walls of plants are made out of cellulose. It's the stringy stuff that's inside of celery. Very hard to digest. In fact, the only thing on the planet that can digest it are bacteria. So cows and things that eat plants, they have bacteria inside of their stomach that break down that cellulose for them, which is why we have a really hard time eating things like corn. It just passes through us because we don't have bacteria, and we can't break down cellulose ourselves. Only bacteria can. Once again, that size difference, much, much, much larger. You guys remember the first page, the prokaryotes about like that, and the eukaryote's huge. So my mouse would be the prokaryote, and the eukaryote's right next to it. And then they have a totally different type of ribosome. It's slightly larger, called an 80S. And then there's kind of a cell type that's in between called a protist. All protists are technically eukaryotic. They have some very simplistic prokaryotic features. They kind of they lack membrane-bound nucleus. Some of them do, some of them don't. Membrane-bound organelles. Some of them do, some of them don't. They're very strange. They're primitive to eukaryotes in some trains of thoughts. They're just they're ancestors to eukaryotes. It just depends on who you talk to. But some of them can cause diseases in people, and a lot of them cause diseases in animals. So it really just depends on the train of thought that you follow, the book that you read, or the person that you're talking to on that one. So prokaryotes. Let's talk about prokaryotes, since this is the main bulk of the class is microbiology. Prokaryotes can be broken up into two giant groups called domains. Now there's 3 different domains of life, and domains are essentially like saying train, boat, and airplane. 3 different categories, large categories of things that move around and take us from place to place. Now, you guys all know that there are tons of different types of cars, tons of different types of trains, and tons of different types of boats. But you guys get the idea. There's way smaller categories underneath the word plane. And that's exactly what here. Domain is just the word plane. You have to further subdivide everything. So, the first division of domain is called eubacteria. Once again, eu means true. Bacteria meaning bacteria. So true bacteria, things that are like Salmonella, E. coli, things that you have heard of. So you look down here, lots of different ones of them all over the place. Gram-positive bacteria, spirochetes, cyanobacteria, the guys that are photosynthetic. Tons and tons and tons of different types of those on the family tree. These guys are the ones that you can probably name by name and think of, and these fit the stereotypical idea of a prokaryote that we defined earlier. The other one is archaea. That's the other domain of prokaryotes. Archaea, archaeology, ancient, old. These guys are organisms that at one point in time were thought to be found, organisms that were found on very primitive, ancient, early Earth. And the reason for that is the fact that they're found in all these terrible environments. So this is down here is called the Big Yellow or Big Blue Hole in Yellowstone National Park. It's boiling hot. It's got sulfur inside of it. It's got a horrible pH. All this yellow color is sulfur. It's full of horrible places that you and I would die if we came in contact with it. Animals die if they come in contact with it. But tons and tons of these archaic organisms live there. It's thought that these guys were, once again, the first forms of life to originate on the planet. It was thought that early Earth had an environment that was very similar and horrible to this. So these guys evolved to live in that horrible place. Now, that's not necessarily the accepted train of thought so much anymore. It's thought that they just specialized to live in these places. You can find archaea all over the place. In the same places you can find bacteria. They live all over the place. Archaea live all over the world, all over pretty much every single place on the planet. So do bacteria. They can pretty much be found right beside one another. Archaea can just be found in some places that bacteria don't like to live so much. Now, the difference between the two, other than the fact that it's blatantly obvious that their environments are different, is they are adapted differently. And this makes perfect sense if you think about it. I like to live in the nice, warm environment that's not too hot, not too cold. It's nice and easy to live in. These guys live in essentially a boiling hot pit with flaming acid. If you live in a boiling hot pit with flaming acid, you have to be able to have your body survive in that boiling hot pit of acid. So that's essentially the differences. The adaptations, the differences in their cell walls, their DNA, their proteins, that allow them to still function in those horrible environments. And that's kind of the difference between the two. They're pretty much the same, but they do differ in some key characteristics, and that key characteristics is what allows these guys to live on the normal, happy environment, and then what allows these guys to live in some sort of horrible hellscape. So let's talk about some of the different eukaryotic members of the microbial world that you might not see as often. So as a microbiology course, we're going to focus more on the bacteria side. We'll talk about these guys a little later on, though. You'll talk about fungi, and you might see a lot of fungal infections, things like athlete's foot, jock itch from time to time. This more severe fungal infections are not as common. So this is fungus down here. This is yeast, budding yeast, little baby bud yeast coming off that's an adult cell. So make alcohol for us, yeast infections, all kinds of crazy stuff. Fungus do all kinds of weird things for humanity. You have algae here, very, very, very cool little species of algae. I really like algae. They come in really different colors, really interesting shapes and textures and things. Some of them are very, very, very dangerous, very toxic for humans. You got your protist here, little ciliate. You can see these little cilia, little swimming around thing, big vacuole here. These guys can time to time cause things like brain-eating amoeba, horrible diseases in humans. Very interesting from time to time. And then you have your tapeworms up here, flatworms, helminths, and things like that. Different types of worms, parasitic worms that get in humans. This is his little mouth here and his little barbs that he uses to stick into your intestinal wall. So there's also a couple of things that are not considered eukaryotes or prokaryotes. They're not even considered living. That are lumped into the study of microbiology. And these are going to be things like viruses, viroids, and prions. So viruses are over here. You guys are familiar with a virus. Things like the flu, you guys, smallpox, you guys have heard of the normal common cold and things like that. And essentially all this is, as you guys can see, it's a big ball. That big ball is made out of proteins. And inside of that is a little tiny piece of DNA or RNA. Now, if you guys recall, our cell theory states that cells have to have both DNA and RNA. Viruses have one or the other. They are not a cell simply because they fail that one test. They don't have both. They fail right out at the gate. They are not a cell. So they have a little protein coat around them with a little piece of genetic material inside, and that's all they are. A viroid is a piece of RNA, just a little raw piece of RNA floating around in the environment. They're kind of shaped kind of like a bobby pin, like a hairpin kind of thing. You guys kind of get the idea. Little hairpin. And these things mostly infect plants. And then a prion down here is going to be a protein that for some reason fails to form properly, or it breaks and it misfolds, it's misshapen. And when that occurs, they don't function properly. They lose their ability to control themselves, regulate their division, and they cause other proteins to misfold as well. They're infectious. And they leave holes in your brain as the proteins start to abandon their natural structure, what they're supposed to do. They leave, and then they infect other proteins, and eventually they cause death by that way. So a little bit of size of the microbial world. Down here we have atoms, smallest thing on the planet, lipids, proteins, flu virus, mitochondria, and bacteria. And if you guys notice, these are about the same size. Very key characteristic here. We'll talk about that later. Animal cells, plant cells, eggs, and things like that, and then an adult human. So ostrich eggs are one single cell. So are chicken eggs. Eggs are one cell, by the way. So this is the largest cell on the planet. So you guys can see the difference between the average size virus and then this one about an average size bacteria. So keep in mind these are the same size, mitochondria and a bacteria. A bacteria is essentially just a mitochondria, and a mitochondria is a bacteria. So keep this in mind. We'll talk about this concept later. So how do we talk about the microbial world when you want to talk about different species? So quick little game real quick to show you why using scientific names is such a big deal. Now in science, you guys have probably heard things like Homo sapien, Canis familiaris for dog, that weird system of names using two different science names, science and Latin names. So system of nomenclature, it's called binomial nomenclature, two different names. So two different names to identify a genus and a species. So why do we do this? Oh, this is a robin. This is an American robin. This is a European robin. They're both birds. They don't look very similar, other than the fact that they're kind of, they kind of sort of vaguely resemble one another. This guy's really tiny, about the size of a little finch. This guy's really large. You guys have seen them in your yard. They're not related to one another, other than the fact that they're both birds. They're kind of like eighth cousins. This is a killer whale. He's not a whale. He's a dolphin. His name implies he's a whale. He's related to porpoises. Orcas tell you nothing about it. Once again, this name robin implies they're related, but they're not. Crayfish, crawfish, crawdad, yabbies, mudbugs. I don't know what they want to call these things. And then buttercups, daffodils, things like that. So lots of different common names that imply information that's not technically right, implies relations that are not there. And then what is a yabby? Well, yabbies are what they call crayfish in Australia. So if you're not in Australia and you don't know what the word yabby is, you're not going to know what this is. So common names differ from place to place. They, once again, you guys can see the yabby example. They differ from tongue to tongue, language to language. I doubt we could speak a different language and know what they were saying. Maybe Alaskan language, native Alaskan language for the word orca. I doubt we know what that was. But if you're an ornithologist that studies birds and someone says to you, I'm going to talk to you about Turdus migratoris, which is the American robin right here, you're going to know, oh, it's not this guy. It's not Erythrica rubelicus, the European robin. It's a different robin. So why does this matter? Once again, it makes things easy. If you're American, you're talking about the American robin. If you're European, you're talking about the European robin. And neither of you have clarified which robin you're talking about if you just say the word robin. Just outright saying the word scientific name, its scientific name, Turdus migratoris, clarifies which one you're talking about. This guy clarifies it's which species of crayfish, crawdad, mudbug, which one it is, because there's a million of them. Over here, gets rid of the confusion of porpoise, things like that. Down here, you get rid of daffodil, buttercup, things like that. And it allows people that don't speak the same languages to talk to one another as well in science terms. Russian, blah, 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 Russian word, blah, 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 Russian word, Turdus migratoris. Oh, and hey, now I know what word you're talking about. I don't know any other thing that you said, but I know what species of birds you're talking about if you don't speak the same language. So when we're talking about bacteria, when we're going to name bacteria, you guys get a little lucky. Bacteria don't really have common names. Their common names are their scientific names for the most part, which is kind of cool. Disease names are different, but the name for the bacteria itself is kind of cool. So Escherichia coli, the genus, anything that's named Escherichia is going to be related to one another. Coli is just the particular species. So it's kind of like saying my last name is Martin. Anybody in my family that has the last name Martin is going to be related to me, but my first name is Jonathan, and that makes me unique to the Martin, Jonathan, Martin, not just a generic Martin. So this is the Martin part, this is the Jonathan part. So this makes it the unique coli species. So how you do this, when you write these, you're going to write them and underline them. So you capitalize the first letter of the genus, the E here. You lowercase the first letter of the species with a space in between. So uppercase genus, lowercase species, space in between. And when you write them, you're going to underline them. So you guys can see here, underline it. When you type them, you can italicize or underline. It's kind of hard to write in italics. So just underline when you write. Now, this really does matter. It seems kind of trivial, but it does matter. There are species of bacteria out there that are also the names of the diseases that they have and also the name of the shape. So Salmonella is also the name of the disease that it causes. Bacillus is the name of a species, the name of a shape. So if you don't specify which one it is, if you don't underline or italicize, which one are you talking about? Are you talking about the species? Are you talking about the shape? Are you talking about the genus itself? Are you talking about the color? I don't know. So you have to specifically underline and specify which one you are talking about. So by underlining and italicizing, it allows someone to know, oh, you're speaking about the species of bacteria and not the shape of bacteria. And that's a big difference, especially if you're talking about, oh, he has an infection by a shape. That sounds kind of strange. So underlining and italicizing really does matter. You can also abbreviate, and you will see it abbreviated from time to time, the names of bacteria. So E. coli, Escherichia coli, S. typhimurium, Salmonella, R. rickettsia is the disease bacteria that causes Rocky Mountain spotted fever. Now, when you do this, you have to be very careful. S. Salmonella, Serratia, Shigella, Staph, Strep. Which one is it? Lots and lots and lots of different bacteria begin with the genus S. So you have to be careful on that one so you know exactly what you're talking about. So we're going to end this little bit of a lecture off on some of the common terms that are found in microbiology. These are very common terms that you guys might see from time to time in clinics, or I might throw out from time to time in class that you probably aren't so familiar with. So let's go ahead and go over these very quickly, and you'll get familiar with what these ideas are. So we've already talked about the first one. What is a microbe? Something that's just too small for you to see with your naked eye. Can't see it. You have to have some sort of device, a microscope, a magnifying glass, to be able to see these particular organisms. The next one is an infection. So what is an infection? So it's the presence and living of another living organism on or in another living organism. So we are infected with tons of different species of bacteria. They live all over you. They live in you. They live on you. Without them, you would die. They are very, very, very important for your survival. They are not causing you disease. They just live on you. So simply because something lives on you and it infects you does not mean that it is necessarily bad. You just have something living on you. You are infected, but it does not necessarily mean that it is bad. What is a sign? Now, you guys will hear the term signs and symptoms from your doctors or healthcare providers and things. So a sign is something that you, as a doctor, a nurse, or whatever, directly observe from your patient. So you look at them, you can see that they have a big giant bruise on the side of their arm. You can see that they have a 98.7 degree fever, normal fever. You can see that they're oozing pustules out of their eyes and things like that. You can observe these things, physically observe them. And this is something that's very useful for you. You can write what you see down and compare these symptoms to charts to figure out what it is, or to other people to figure out what a new disease is, something that you physically observe from your patient. Now, symptoms are something that your patients physically tell you. My stomach hurts. Oh, it hurts when you touch that. I have a headache. It feels weird here. Things that they tell you. Now, they are not doctors, for the most part, usually not as often. They might not be as well trained in the healthcare field. So sometimes you have to take symptoms with a grain of salt. My tummy hurts. Well, if you have ever talked to a little child, their tummy ranges from the bottom of their rib cage to the top of their belly button, or the bottom of their belly button. So that's a rather large way that contains lots of different organs. Which one of them hurts? So that's not necessarily as useful. So inoculation. You'll use this term a lot in labs. You'll use this term sometimes in vaccination talks. And that's essentially just introducing a brand new species or culture of bacteria into some place where it's never been before. So a brand new environment. So we inoculate a petri dish, we inoculate a test tube, you can also inoculate a patient and things like that. So disease. What is a disease? You got your foot run over by a car, it crushed it, it hurts. That is not a disease. That is tissue damage that's caused by a physical injury. You got your foot run over by a car, you were healed, and then your cut got infected with gangrene and your foot had to be amputated because it fell off from all the toxins. That is a disease. Tissue damage with a root cause that is not a physical injury. You can go, oh, there is an organism there that is causing that foot to fall off. I can see it. I know what it is. I can say it is not because of the car wreck that that person's foot fell off. It is because of the bacteria that came after after their injury got infected. That's what a disease is. When an infection crosses over into physical injury, it's a disease. You have to have a living organism that causes physical injury and tissue damage. Or excuse me, a living organism that causes tissue damage. That's a disease. So septicemia, you guys have heard this term a lot before. A lot of crime scene shows use it a lot. Oh, he died of septicemia. That's when bacteria and or their toxins are found inside of your blood, actively growing and dividing. They're doing their thing. They're happily growing and dividing, just living, doing their thing in your blood, making more of themselves. And they also have toxins in your blood, and or it could be either. Toxins in your blood are not a good thing. It can be moving around from organ to organ, muscle to muscle, location to location throughout the bloodstream, causing damage throughout the way. The bacteria can spread from location to location, muscle to muscle throughout the body and cause problems as well. So bacteremia is just the presence of bacteria in your blood. Not necessarily a bad thing to have bacteria in your blood. It's not a good thing. They're not actively dividing, they're just there. It's not a great thing, not necessarily a bad thing, just something you want to be aware of. If they're actively dividing, growing, and doing their thing in your blood, and their toxins are there, or their toxins are there, you have problems. Septicemia is bad. Bacteremia is not necessarily bad, but it has the potential to go bad very quickly. So it's just something to be aware of. And then the last little bit here. What is a parasite? It's an organism that lives on or in you that feeds off of you. Pathogen is an organism that is a microbe that is designed or evolved to do harm. So you have two different types of pathogens. You have something called an obligate pathogen or an opportunistic pathogen. But first, I'm going to talk about what these two things here are, and we'll come back to this. So normal flora. You have bacteria that live on and in your body. They make up about 5 to 10% of the average person's body weight. It takes about a day or two for them to colonize you once you're born. You're born sterile. You shouldn't have any bacteria on or in the womb. You're born, they get on you from the birth process, from the environment, then they start to grow on you. They're very important for your survival. You have to have them to be healthy. Without them, you would die. The bacteria that live on and in your body, for the most part, don't cause you harm. They need you to live. You need them to live. If they kill you, it's bad. They don't want to do that. So they live on you. They do their thing. They don't cause you any problems at all. These bacteria are called normal flora. They keep you happy, they keep you healthy, and you do the same for them. Now, occasionally they can cause you problems. If you have something called a staph infection, you guys are familiar with that one, that can be caused when a normal flora bacteria, one that normally doesn't cause you any problems, can cause problems. So you get a cut, the bacteria that are living on your skin, the staph, go, oh, that's a nice, warm, moist place that's got a lot of food inside. I'm going to move in. And they move in and they start causing problems. And that's something called an opportunistic infection. That bacteria never would have caused problems to you had the opportunity never arisen for it to do so. Had you never cut yourself, you would have lived the entire rest of your life without the normal flora of your body causing you problems. That staph is not going to cause problems anywhere else on your body unless you cut yourself. It is a pathogen of opportunity, an opportunistic pathogen. Now, a true pathogen, on the other hand, is a bacteria that causes problems no matter what. If it lands on your normal flora, if it lands on you at all, if it gets on or near you, no matter what, it will cause you problems. You cannot escape this one. That is called a true pathogen. Normal flora live on you at all times. If a pathogen lived on you at all times, you would be dead. So normal flora are good bacteria that sometimes do bad. Pathogens, true pathogens are bad bacteria that always do bad. So that's the difference between a true pathogen and an opportunistic pathogen. So the last little bit of this lecture, I'm going to post a video online for you guys on YouTube of what this stuff is. This is not going to be something that I'm going to harp over too much. We will talk about this a little bit later on in the course. So I'm going to post a video that gives a little better explanation of this with some more visual pictures and things, a little more animation. So this would be a little more explanation on the video. So go ahead and check that out. So this is going to be the end of the lecture for me. If you guys have any questions at all, please feel free to contact me about it. If not, have a good rest of the day.